Assalamu alaikum طلاب, and welcome to the second lecture of the organs associated with the digestive tract. Today's lecture will be of two parts. The first part we will discuss the histological structures of the biliary tract and gallbladder, and the second part will be we will discuss the histological structure of the pancreas with its two main parts, the endocrine and the exocrine part. So starting with the biliary tract and gallbladder, the objectives. We have three main objectives. So the first one is to identify and describe the histological features of the intra-hepatic biliary passages, and we should identify and describe the histological features of extra-hepatic bile ducts, and finally we will discuss and describe the histological features of the gallbladder. As you know from the previous lecture, the bile is produced by hepatocytes. After its production from the hepatocytes, bile will flow into a number of canals or tubes, starting from bile canaliculi and ending in the common bile duct before its excretion into the duodenum to aid in the process of absorption of fat from the duodenum. We will see and discuss the histological structure of each one of them and know them in details. So starting with the bile canaliculi, as you know from the last lecture, the previous lecture, bile canaliculi is the smallest branch of the biliary tree. Bile canaliculi is the smallest branch of the biliary tree, and it's formed by the opposed grooves in the surface of adjacent hepatocyte. As you can see here, we have two hepatocytes on each side of the bile canaliculi, and on this side we have a groove that by exact opposition of the hepatocyte a tube will be formed called bile canaliculi. So the wall of the bile canaliculi is formed only from one type of cell, that's the hepatocyte. We have another view of the bile canaliculi after staining of the liver tissue with a special stain. You can see here this black lines represent the bile canaliculi present between two hepatocytes. These bile canaliculi will then form the canals of herring, where the lumen of these canals is formed from two types of cells, hepatocytes and cholangiocytes. The cholangiocytes is the main cell that form the lining epithelium of the biliary tree. So here is the connection between the bile canaliculi and the canals of herring. These cholangiocytes will gradually increase in number until finally the bile passage is only lined with cholangiocytes, and the structure is known now as intra-hepatic bile ductule. Intra-hepatic bile ductule. This ductule is about 1 to 1.5 micrometer in diameter, and as I said, it's lined entirely by cuboidal or sometimes columnar cholangiocytes. These bile ductules will merge together, forming the bile ducts. The bile ducts, as we said in the previous lectures, are located within the portal area at the periphery of the hepatic lobules. Bile ducts are about 15 to 40 micrometer in diameter, and it's just like the bile ductules, only lined with cholangiocytes. And we have a longitudinal section of a bile duct, and you can see clearly the cholangiocytes lining its surface or lining its lumen. These are, as we said, the hepatocytes in the liver lobule. Again, we have this liver lobule, and on the liver lobule we have the portal area. The portal area, as you know, is formed from a branch of portal vein and a branch of hepatic artery and bile duct. Bile ducts merge together, forming the right and left hepatic ducts. right and left hepatic duct, and we can see this clearly in anatomy. These two will fuse together, forming the common hepatic duct, which will join the cystic duct, forming the common bile duct. The common bile duct will open in the duodenum in the ampulla of water. While the portal duct in the portal area is lined with the cuboidal to locoruminal cholangiocytes, we have the two hepatic ducts, cystic duct, common hepatic duct, and the common bile duct are lined with a mucous membrane having simple columnar epithelium formed of cholangiocytes with thin lamina propria and thin submucosa. We have some mucous glands in some areas of the cystic duct. These are surrounded by thin muscularis layer, and this muscle layer become thick near the duodenum finally when it opens, forming the duodenal papillae, so it forms a sphincter that regulates bile flow into the small intestine. Again, this is a longitudinal section of a bile duct in the portal area, and we can see here the cholangiocyte, that these are cuboidal to locoluminar cells lining its lumen. The gallbladder is a hollow pear-shaped organ, as we can see it here in this photo. Is a hollow pear-shaped organ attached to the lower surface of the liver, and it's capable of storing 30 to 50 ml of bile that's concentrated during storage. The wall of the gallbladder is composed of three main layers. It has a mucosa, a muscle layer, and a serosa. The mucosa, as we can see here, is the first layer lining the lumen of the gallbladder, and it consists of simple columnar epithelium, and its main function is to absorb water from the bile and concentrate bile. Beneath the surface epithelium, which is simple columnar, we have a lamina propria of loose connective tissue, which typically exhibit numerous deep folds in the mucosa, and these deep folds especially evident where the gallbladder is empty or partially full. The mucosa will be flattened when the gallbladder is full. Beneath the mucosa, we have a thick layer of muscle coat. The muscle cells here is oriented in all the direction irregularly, and as we see here, there is no muscularis mucosa or submucosa in the gallbladder, so we have the mucosa, and directly beneath it is the muscle cell. Contraction of muscle here in this coat will cause emptying of the gallbladder content, so release of bile into the small intestine. External to the mucosa, external to the muscle coat, we have the adventitia containing adipose tissue and a blood vessel. And as we said, the main function of the gallbladder is to store bile produced by the liver. It also serves to concentrate the bile through reabsorption of large quantities of water and mineral salts through its mucosal layer, and emptying the concentrated bile into the common bile duct. This photos is taken from the histology manual, histology guide, that you use it for studying practical histology. And as we can see here, the wall of the gallbladder is composed of three layers, namely the mucosa, muscle layer, and serosa. Mucosa, muscle layer, and serosa. Here is an enlarged micrograph of the mucosa, and we can see clearly the tall columnar cells lining the lumen of the gallbladder, the lamina propria, and it consists of loose connective tissue, and below the lamina propria we do not have a submucosa or muscularis mucosa, but immediately we have a thick layer of muscular coat with muscle cells arranged in all directions, and the external layer is the serosa or adventitia. Part 2 of our lecture is the pancreas, And we have several learning objectives in this part. First, we need to discriminate between the endocrine and exocrine pancreas, discuss the cellular structure of the exocrine pancreatic acinus and its function, identify the ductal system of the exocrine pancreas, and identify the intercalated duct, centroacinar cells, interlobular and intralobular duct, recognize the endocrine part of the pancreas, which is the eyelids of Langerhans, and discuss the various types of cells within this eyelids of Langerhans. So to start with, a few words about the pancreas. We all know that the pancreas is a mixed endocrine and exocrine gland. That's to say, it produces both digestive enzymes and hormones. It's an elongated retroperitoneal organ with a head, body, and tail region. The pancreas has an ill-defined fibrocollagenous capsule that sends septa to the inside of the gland, dividing it into irregular lobules. The majority of the pancreas is an exocrine part that form about 99% of the pancreas. The cells of this exocrine pancreas secrete a mixture of fluid and digestive enzymes, and collectively they are known as pancreatic juice, such as proteases, amylases, lipases, nucleases, different digestive enzymes. The proteases are secreted as inactive form, such as trypsinogen, chymotrypsinogen, and others. This pancreatic juice is characterized by being extremely alkaline, and the pancreas roughly secrete about 1.5 liter of pancreatic juice per day and deliver it directly to the duodenum, where it neutralizes the acidic chyme entering the duodenum from the stomach. And also this alkaline pH is very important for optimal activity of the pancreatic enzymes. The exocrine pancreas is formed from acinar cells, the same as acinar cells that we see in parotid gland. So the pancreas and the parotid gland are very similar in this feature. It's very clear from this photo that the pancreas is surrounded by a connective tissue capsule that sends septa to the inside of the pancreas, dividing it into an irregular lobules. These lobules are formed mainly from clusters of acinar cells, clusters of serous acini. These serous acini are formed or consist of several pyramidal serous cells, as we can hear. This is an acinus, serous acini, and they are formed from several serous cells. These serous cells are pyramidal in shape with a broad base and narrow apex, surrounding a very thin lumen, very small lumen, and these are surrounded by a thin basal lamina and supported by a delicate sheath of reticular fibers which is rich in capillary network. If we want to describe these cells, we can say that, as we said earlier, it's pyramidal shaped cells with a narrow apex and a broad base. The cells have a rounded basal nuclei. The cells are rich in rough endoplasmic reticulum that is concentrated mainly in the lower half of the cell, and it's responsible for their cytoplasmic basophilia. In the apex of the cell, closer to the lumen, we have variable number of eosinophilic zymogene granules, as we can see here. It's very clear. These granules contain the pre-enzymes synthesized by the cell. So if we better to see the appreciation of the acinar cells or pancreatic acini under high power microscope, we have this photomicrograph. So this is an acinus. This is an acinus. We have these pyramidal acinar cells. We have rounded, basally located nucleus, and we have cytoplasmic basophilia in the lower part of the cell from the abundant rough endoplasmic reticulum, and we have numerous eosinophilic zymogene granules in the apex of the cell. And as we said, this is a acinar, pancreatic acini. Electron microscope will show the same features. So we have these acinar cells, we have the rounded nucleus, basophilia, and we have here the zymogene granules very evident near the lumen. These zymogene granules will secrete the pre-enzymes into the lumen of the acinus by a process of exocytosis. The secretion of these acini, or the pancreatic juice produced by this acinus, for example, will be collected by intercalated duct. The smallest duct in the pancreas is called intercalated duct, and as we remember in the parotid, we have the smallest duct in the parotid gland, also called intercalated duct. The initial cell of these ducts within the acinus is called centroacinar cell. These are small, pale, staining cells with a small nuclei, and they represent the first cell of the intercalated duct that is within the acini. These cells are unique to the pancreas. They can't be found in parotid, for example. They only found in the pancreas, and they have a centrally placed flattened nucleus with an attenuated cytoplasm. That's to say, they are squamous in shape. They are not cuboidal or columnar, however, they are squamous in shape. And these cells are continuous with the cells forming the intercalated duct. The intercalated duct, as we said, lies outside the acinus. The duct system of the exocrine pancreas, we can say that it starts with the centroacinar cell. It starts with the centroacinar cell, and these are the centroacinar cells, and then the intercalated duct. The intercalated duct will merge together, forming an intralobular duct, which merge together forming interlobular duct, and these lobular duct will merge together forming the main pancreatic duct. So if we look at these structures within the histologyguide.com, we can see that this is a section from the pancreas showing collection or spherical collection of cells, of serous cells, known as pancreatic acini. Between them we can see a very small duct that is known as intercalated duct. The larger duct, but still within the lobule, is called the intralobular duct. However, if we move outside the lobule within the connective tissue, between the lobules, we can see the interlobular duct, and many of these interlobular ducts merge to form the main pancreatic duct. The last part of today's lecture is the endocrine pancreas, the histological feature of the endocrine pancreas. While we said the exocrine pancreas form about 99% of the organ, this means that the endocrine pancreas forms only 1 to 2% of the organ's total volume. However, we still have large number of eyelids of Langerhans within the pancreas. So we have about between 1 to 3 millions of eyelids of Langerhans. They are concentrated mainly in the narrow tail region, and we can see it under light microscope as a rounded or oval structure composed of cells. This eyelids is about 100 to 200 micrometer in diameter, and they are scattered throughout the pancreatic tissue and embedded within the exocrine acinar part of the pancreas. A very thin reticular capsule surrounds each eyelid, separating it from the adjacent acinar tissue. The cells within the eyelids of Langerhans are as you can see here, are polygonal or rounded. They are smaller and more lightly stained than the surrounding acinar cells. They are arranged in cords separated by fenestrated capillaries. And this fenestrated capillary has contact with every cell within the eyelids of Langerhans. Although it's not evident with light microscope, but with electron microscope, these cells have all features of active polypeptide secreting cells. So they have a well-developed rough endoplasmic reticulum, a prominent Golgi, and very numerous secretory granules within its cytoplasm. Immunohistochemically, a special type of staining divided the cells within the pancreatic eyelids into 4 groups. The most common are beta cells, which form 70% of the eyelids, and they are concentrated in the center of the eyelids. The importance of these cells is that they secrete insulin, and as we said, they are concentrated within the center of the eyelids. The other type of cell is alpha cell, and they secrete glucagon, alpha cell, and they secrete glucagon, and they are found in the periphery of the eyelids. Delta cell comprise about 5 to 10% of the eyelids of Langerhans cells, and these cells secrete somatostatin. They are much less abundant, and they are found within the other types of cells. And we have a special type of cell called pancreatic polypeptide cell, and they comprise only a few cells, about 1 to 2% only of the number of cells within the eyelids, and the importance of these cells is that they secrete pancreatic polypeptide. This is a schematic diagram of the eyelids of Langerhans. Here the pancreas is stained with a special stain called Mallory-Azan method, and by this method we were able to identify 3 of the 4 types of cells within the eyelids of Langerhans. So we have A cell that display red cytoplasmic staining, and as we said, it secretes glucagon. We have beta cell, which comprise most of the eyelid cells, and they display brownish orange staining. And we have delta cells that has a blue cytoplasm and secrete somatostatin. And by this we reach to the end of our. Thanks a lot for your listening.